everyone, welcome back. We're doing a deep dive today into something kind of mind-blowing. Yeah, the potential of ivermectin as an anti-cancer drug.
Interesting.
Specifically for colorectal cancer.
Huh.
You might know ivermectin as that common antiparasitic medication.
Right.
But the research we're going to be looking at today reveals that it may have some hidden talents.
Okay, cool.
So for this deep dive, we've got two sources.
Okay.
A research article from Frontiers in Pharmacology titled, Ivermectin has new application in inhibiting colorectal cancer cell growth.
Wow.
And a review article from PMC called Ivermectin, a potential anti-cancer drug derived from an antiparasitic drug.
Wow, it's fascinating how research is always uncovering these new possibilities for existing drugs, you know?
Exactly.
Sometimes it feels like we're just scratching the surface of what these medications can do.
This is a perfect example of that.
Yeah.
So before we get into the specifics, could you give us a quick rundown of ivermectin's traditional use?
Sure.
And how it's been making waves in other areas lately?
So ivermectin has been a crucial tool in fighting parasitic infections for decades.
Right.
It's even on the World Health Organization's list of essential medicines.
Oh, wow.
Which just speaks to its global impact.
Yeah.
In recent years, it's also been explored for potential antiviral properties.
Oh, really?
But that's a whole other deep dive for another time.
Okay. I'll add that to the list.
Sounds good.
But for today, we're really focusing on ivermectin's potential in the battle against cancer.
Right.
And the Frontiers in Pharmacology article dives into some pretty exciting findings on its effects on colorectal cancer cells.
Yes. And they focus on two specific types of colorectal cancer cells.
Okay.
SW480 and SW1016.
Got it.
Studying different subtypes like this helps researchers understand if a drug's effects are broad or specific to certain types of cancer.
That makes sense.
Yeah.
So what did they find? Was ivermectin effective in stopping these cancer cells from growing?
The results were really compelling.
Okay.
They found that ivermectin effectively inhibited the growth of both SW480 and SW116 cells.
Oh, wow.
And what's really interesting is this inhibition was dose-dependent.
Dose-dependent?
Yes.
Meaning the higher the dose of ivermectin, the more it stopped the cancer cells from growing.
Exactly.
Yeah.
This is a common finding in pharmacological studies.
Right.
And it helps researchers figure out the best dosage for potential treatments.
Okay.
The study even went a step further and looked at what's called the IC50 value.
Okay.
This tells us the concentration of ivermectin needed to inhibit 50% of cell growth.
So getting into the nitty-gritty now.
Yes.
What were those IC50 values?
Well, for the SW480 cells,
the
IC50 values hovered around 16 sero at 12 hours.
Okay.
About 15 sero at 24 hours.
Okay.
And 12 VM at 36 hours.
Got it.
But for the SW1116 cells, those values were notably lower.
Okay, interesting.
Around 7.6 BIM at 12 hours.
Wow.
6.3 AM at 24 hours.
Right. And 5.8 NATO at 36 hours.
So a lower dose of ivermectin was needed to achieve the same level of growth inhibition in the SW1116 cells compared to the SW480 cells.
Right.
What does that tell us?
Well, it suggests that the SW1116 cells are more sensitive to ivermectin.
Interesting. Why would there be a difference?
It could be due to genetic or molecular differences between the cell lines.
Okay.
But that would require further research to pinpoint.
Got it.
It's definitely a clue worth investigating further.
It's like each cell line has its own personality and reacts differently to the same medication.
Yeah.
Makes you wonder if these differences could be harnessed for more targeted therapies in the future.
Absolutely. That's the direction personalized medicine is moving towards.
Right.
But getting back to ivermectin's impact on these cancer cells, the Frontiers in Pharmacology article also included some pretty striking images.
Oh, cool.
Of what happened to the cells after ivermectin treatment.
I love when research papers include visuals.
Me too.
It helps to really grasp what's going on.
What did the images show?
Well, as the ivermectin concentration increased, the cells started to shrink, become rounded, and even detached from the surface they were growing on.
Those sound like pretty drastic changes.
They are.
What was going on at the cellular level to cause these changes?
Well, those morphological changes, as they're called,
Okay.
strongly suggest that the cells were dying.
Okay.
And not just randomly. The research points toward ivermectin triggering a specific process called apoptosis.
Apoptosis?
Yes.
Isn't that like programmed cell death?
You got it.
The cell's self-destruct sequence?
It's a crucial process for maintaining healthy cell populations.
Yeah.
In a way, it's the body's way of getting rid of cells that are damaged or no longer needed.
Right.
And in the context of cancer,
Yeah.
inducing apoptosis in those rogue cells is exactly what you want to happen.
So how did the researchers confirm that ivermectin was actually causing apoptosis?
They used a technique called flow cytometry to analyze the cells after ivermectin treatment. This technique allows them to see specific markers that indicate apoptosis is happening.
Got it.
And the results were pretty clear.
Okay.
The higher the ivermectin concentration, the more cells were undergoing apoptosis.
That's pretty convincing.
Yeah.
But apoptosis is a complex process.
Right.
Did they figure out what was happening inside the cells to trigger this self-destruct sequence?
Yes. They dug deeper and found that ivermectin was increasing the activity of an enzyme called caspase-3.
Caspase-3.
You could think of caspase-3 as the executioner in the apoptosis pathway.
Okay.
When it gets activated, it sets off a chain reaction that ultimately dismantles the cell.
So ivermectin is boosting caspase-3.
Yes.
Which is basically flipping the switch on the cell's self-destruct button.
Right.
But there are multiple pathways involved in apoptosis, right?
There are.
Did the study narrow down which one ivermectin was activating?
They focused on what's called the intrinsic pathway of apoptosis.
Okay.
This pathway is regulated by a group of proteins called the BCL2 family. Some of these proteins, like BAX, promote apoptosis, while others, like DCL2, try to prevent it.
So it's like a tug of war between these proteins determining the fate of the cell.
Exactly.
Where did ivermectin fit into this struggle?
Ivermectin seemed to tip the scales in favor of apoptosis.
Interesting.
The study found that it increased the expression of BAX, the pro-apoptosis protein, and decreased the expression of BCL2, the anti-apoptosis protein.
So it shifted the balance.
Yes. This shift in balance ultimately disrupts the mitochondria, which are crucial for cell survival.
It sounds like ivermectin is playing a strategic game inside the cell.
It is.
Manipulating these key proteins to achieve the desired outcome.
And there's even more.
Oh, really?
They also looked at the impact of ivermectin on a protein called PRP.
Okay.
This protein is involved in DNA repair, but during apoptosis, it gets deactivated.
So if KIRP isn't working, the cell can't fix its DNA, and it's even more vulnerable to cell death.
Exactly, and this study showed that ivermectin treatment did indeed lead to increased levels of deactivated PRP.
Oh, wow.
Further supporting its role in triggering apoptosis.
So ivermectin is really going all in on this apoptosis strategy.
It seems so.
I'm curious, did the research explore any other ways in which ivermectin might be killing cancer cells?
You're right, there's more to the story.
Oh, good.
They also investigated something called ROS, or reactive oxygen species.
Now remind me, what are ROS again?
They're basically byproducts of cellular metabolism.
Okay.
Think of them like exhaust fumes from the cell's engine.
Okay.
A little bit is normal, but too much can be toxic and cause damage to the cell.
So did ivermectin increase ROS production in these cancer cells?
That's exactly what they found.
Wow.
They saw a dose-dependent increase in both total and mitochondrial ROS production after ivermectin treatment.
Okay.
And remember how we talked about ROS being able to trigger apoptosis?
Yeah.
Well, that's where things get really interesting.
So ivermectin is boosting ROS, which could be contributing to the cell's demise through apoptosis.
You got it.
It's like a double whammy.
It is.
But they didn't just assume this was happening.
Right.
They wanted to confirm ROS was playing a role, so they used a substance called NAC, which is known to inhibit ROS production.
Exactly.
And guess what?
I'm intrigued.
When they added NAC to the mix, it significantly reduced the ROS accumulation caused by ivermectin.
Wow.
And here's the kicker, it also reduced cell death.
Oh, interesting.
This suggests that ROS is indeed a key player in ivermectin's anti-cancer activity.
Fascinating.
Okay, so we've got ivermectin triggering apoptosis, boosting ROS production, and ultimately leading to the death of those cancer cells.
It's pretty impressive.
It is. But I have a feeling there's even more to this story, right?
You're right on track.
Okay, good.
We've only just scratched the surface. There's another fascinating mechanism at play here.
Oh.
Cell cycle arrest.
Cell cycle arrest.
Yes.
Fill me in on what that means.
So every cell goes through a cycle of events that lead to it dividing and creating two new cells.
Right.
This cycle is tightly controlled.
Yeah.
But cancer cells often find ways to bypass these controls and divide uncontrollably.
Right. That's part of what makes them so dangerous.
Exactly.
So where does ivermectin fit into this cycle?
The researchers found that ivermectin actually caused cell cycle arrest in both the SW480 and SW1116 cells.
Really?
Specifically, it arrested the cycle at the S phase.
Okay, back up for a sec.
Sure.
What exactly is happening during the S phase?
So the S phase is all about DNA replication.
Uh-huh.
It's the phase where the cell copies its entire DNA so that each new daughter cell will have a complete set.
Got it.
If you arrest the cell cycle during this phase, the cancer cells can't replicate their DNA,
Okay.
which essentially stops them from multiplying.
So ivermectin is not only killing cancer cells directly,
Right.
but also preventing them from making copies of themselves.
Exactly.
It's like hitting the pause button on cancer cell proliferation.
It is.
How did they figure this out?
They used another clever technique called flow cytometry,
Okay.
which allows them to see which phase of the cell cycle each cell is in.
Got it.
And the results showed a clear accumulation of cells in the S phase after ivermectin treatment,
Oh, wow.
confirming that the cycle was being arrested at that point.
This is fascinating.
It is.
We've covered a lot of ground already. Apoptosis, ROS, cell cycle arrest.
We have.
It seems like ivermectin is attacking cancer cells from multiple angles.
And we're just getting started.
Oh, no.
There's so much more to explore, including ivermectin's potential against other types of cancers and its possible role in combination therapy.
Okay, I'm officially hooked.
Excellent.
We'll be back in part 2 to delve deeper into these exciting possibilities. Don't miss it.
I'll be here.
Welcome back, everyone. In part 1, we uncovered some pretty amazing things about ivermectin's potential as an anti-cancer agent.
We did.
We talked about how it triggers apoptosis,
Right.
messes with ROS production, and even throws a wrench in the cell cycle.
It's pretty amazing.
But we've only just begun to explore the depths of this research.
It's true. We've barely scratched the surface. Ivermectin's potential extends far beyond just colorectal cancer.
Right.
And there are some really exciting possibilities for using it in combination with other cancer treatments.
Okay, before we dive into that, I want to go back to something we touched on earlier.
Okay.
The difference in ivermectin sensitivity between those two colorectal cancer cell lines,
Right.
SW480 and SW1116.
Yeah.
You mentioned that the SW1116 cells were more sensitive,
They were?
needing a lower dose of ivermectin to inhibit their growth.
Correct. Tailoring treatments to the unique characteristics of each patient's cancer.
That makes a lot of sense. But what could be driving these differences in sensitivity? Is it something inherent to the cells themselves?
That's a question researchers are actively investigating.
Mm-hmm.
One possibility is that these cell lines have different genetic or molecular profiles.
Okay.
They might have variations in genes involved in drug uptake, metabolism,
Okay.
or even those apoptotic pathways we discussed.
So it's like each cell line has its own unique fingerprint
Yeah.
that determines how it responds to a drug.
Precisely. And the more we understand these fingerprints, the better we'll be at developing targeted therapies that exploit these differences
Right.
and maximize effectiveness.
That's really fascinating. It seems like every new piece of research opens up even more avenues to explore.
It does.
Speaking of which, you mentioned that the PMC review article we're using for this deep dive
Yes.
points to ivermectin's potential in combination therapy.
Right.
What exactly does that mean?
Combination therapy is a cornerstone of modern cancer treatment.
Okay.
It involves using multiple drugs that attack cancer cells from different angles.
So instead of just one superhero, you have a whole team of them, each with their own special powers.
I love that analogy.
Yeah.
And the benefit of this approach is that it can be much more effective than using a single drug alone.
It makes sense. If you hit the cancer cells from multiple directions, they have less chance of surviving or becoming resistant to the treatment.
Exactly. And that's where ivermectin comes in.
Okay.
With its multifaceted mechanisms of action-inducing apoptosis, increasing ROS, causing cell cycle arrest, it has the potential to be a valuable addition to this superhero team.
Okay, so what kinds of combinations are researchers looking at? What other cancer-fighting agents are being paired with ivermectin?
The PMC article highlights a few promising combinations.
Okay.
For example, ivermectin has shown synergistic effects with cisplatin.
Synergistic effects, meaning the combination is more powerful than the sum of its parts.
Exactly. And cisplatin is a chemotherapy drug that's used to treat a variety of cancers, including ovarian cancer.
So it sounds like ivermectin might be able to give traditional chemotherapy drugs a boost,
Right.
making them even more effective.
That's one of the exciting possibilities. And the research doesn't stop there.
Oh.
They've also found that ivermectin shows promise in combination with paclitaxel,
Okay.
another widely used chemotherapy drug.
Got it.
And there's even some evidence suggesting it could enhance the effects of targeted therapies like dasatinib,
Okay.
which is used to treat chronic myeloid leukemia.
Wow. It seems like ivermectin can play well with others, regardless of their therapeutic approach.
And that versatility stems from its ability to target multiple pathways involved in cancer cell survival and proliferation.
Right.
It's like having a Swiss Army knife of anti-cancer mechanisms.
I love that image. It really underscores the potential of this drug. Now, I remember you mentioning something about ivermectin's impact on cancer stem cells.
Yes,
Can you tell me more about that?
absolutely. Cancer stem cells are a subpopulation of cancer cells
Okay.
that are thought to be responsible for tumor initiation,
Okay.
growth, and recurrence.
So they're particularly tricky to target,
They are.
like trying to take down the head of a criminal organization.
Exactly. Traditional cancer treatments often target the bulk of the tumor cells.
Right.
But they might miss those sneaky cancer stem cells,
Yeah.
which can then lie dormant and reemerge later, leading to relapse.
That sounds like a nightmare scenario.
It can be.
So how does ivermectin fit into this picture?
Some studies suggest that ivermectin might be able to target these elusive cancer stem cells.
Wait, really?
Yes,
That would be a game changer.
it would.
How is it doing that?
The exact mechanisms are still being investigated,
Okay.
but there's evidence that ivermectin might be interfering with the self-renewal and differentiation capabilities of these cancer stem cells. Essentially, it could be cutting off the cancer's ability to regenerate itself and form new tumors.
So it's not just about killing the existing cancer cells. Right. It's about preventing the cancer from coming back with a vengeance.
Exactly. If we can effectively target those cancer stem cells, it could dramatically improve long-term outcomes for patients.
This is incredible. It seems like ivermectin is revealing new layers of potential with every study.
It is.
But we were also talking about multidrug resistance.
We were.
Can you remind me what that is and how ivermectin might play a role in overcoming it?
Multidrug resistance is a major hurdle in cancer treatment.
Okay.
It happens when cancer cells develop mechanisms to resist the effects of multiple drugs,
Yeah.
making them incredibly difficult to treat.
So it's like those cancer cells are evolving,
Yeah.
finding ways to outsmart our therapies.
Precisely. They might pump the drugs out of the cell before they can do any damage,
Oh, wow.
or they might alter their internal pathways to bypass the drug's effects.
That sounds like a really formidable challenge.
It is.
So how does ivermectin come into play?
There's evidence that ivermectin might be able to reverse this multi-drug resistance by inhibiting a protein called P-glycoprotein, or PGP for short.
Okay, back up for a second. What does PGP do?
Sure.
And why is it such a problem in cancer treatment?
PGP acts like a bouncer at the cell membrane,
Okay,
pumping drugs out of the cell before they can reach their targets.
got it.
Think of it as a defense mechanism that cancer cells use to protect themselves from our medications.
So ivermectin is essentially disarming this defense mechanism,
Yes.
allowing the other drugs to get in and do their job.
That's one way to put it.
That's pretty cool.
And what's really exciting is that this ability to reverse drug resistance has been observed in various cancers,
Oh, wow.
including colorectal cancer, breast cancer, and leukemia.
It's incredible to think that ivermectin could be this multifaceted weapon in the fight against cancer, not only attacking the cancer cells themselves, but also boosting the effectiveness of other therapies and potentially even preventing those pesky cancer stem cells from staging a comeback.
It really is remarkable how research continues to uncover hidden potential in existing drugs.
It is.
Ivermectin is a prime example of the power of scientific inquiry and the importance of thinking outside the box.
Okay, I'm already on the edge of my seat for part 3. We've talked about apoptosis, ROS, arrest combination therapy, cancer stem cells, and multi-drug resistance.
A lot.
What else is there to explore?
We're going to delve deeper into one of the most fascinating targets of ivermectin,
Okay.
a kinase called PAK1.
PAK1.
It's a key player in many cancer-related pathways,
Uh-huh.
and ivermectin's ability to inhibit it could have huge implications for cancer treatment.
I can't wait to hear more about it. Don't miss the final part of our deep dive where we'll unravel the mysteries of PAK1 and explore the future of ivermectin as a potential cancer-fighting hero.
I'll be here.
Welcome back to the final part of our deep dive into ivermectin. Its amazing potential as a cancer fighter.
It's been quite a journey.
It has. We've covered so much already: apoptosis, cell cycle arrest, even combination therapy and cancer stem cells.
It really shows the breadth of research being done on ivermectin.
It does. But there was one more thing you mentioned.
All right.
A kinase called PA1.
PA1 is a fascinating target. You see, it's often overexpressed in cancer cells.
Meaning there's more of it in cancer cells than in normal, healthy cells.
Exactly. And this overexpression is often linked to tumor growth and progression.
So if PK1 is leading the cancer army astray,
Kind of, yeah.
ivermectin steps in to neutralize it.
That's a good way to put it.
Yeah.
By inhibiting PA1, ivermectin disrupts the signals that tell cancer cells to grow, survive, and spread.
So like cutting off the communication lines?
Yes, precisely.
How does that actually work?
Well, one crucial pathway is the actin-MPO pathway.
Okay.
It plays a major role in cell growth and proliferation.
Yeah.
You can think of it as the accelerator pedal in a car.
Okay.
And in cancer cells, this accelerator is often stuck to the floor, driving uncontrolled cell division.
So they're just constantly growing.
Yeah. But ivermectin, by inhibiting PAK1, seems to cut the fuel line to that accelerator.
Slowing down the growth.
Exactly.
That makes sense.
Yeah.
There's also the MAPK pathway, right?
That's another important one. It's involved in cell growth and survival, too, but it's more like the steering wheel.
Okay.
It helps guide the cell's fate,
Interesting.
whether it will divide, differentiate, or even die.
And this is messed up in cancer cells.
Often, yeah. The steering wheel malfunctions leading to all that abnormal growth and development.
So is ivermectin kind of recalibrating the steering wheel then?
You could say that by interfering with those signaling pathways, ivermectin disrupts the cancer cell's command center.
This is reminding me of our talk about ROS,
yeah.
those reactive oxygen species.
Right.
Could ivermectin's impact on TAK1 be connected to its effects on ROS production?
That's a really interesting question.
Yeah.
And it's definitely an area for more research.
Yeah.
While there isn't a clear answer right now, it's possible that those processes are linked in ways we don't fully understand yet.
It seems like ivermectin is attacking from all sides, hitting cancer cells in so many different ways.
It really is remarkable. And that multi-pronged approach makes ivermectin so promising.
Ew.
Because it means the cancer cells are less likely to develop resistance.
Right, if it's attacking from every angle at once.
Exactly.
So we've got this drug that was traditionally used to fight parasites.
Yeah.
And now it might be able to kill cancer cells, stop them from multiplying, make other treatments more effective.
And even target those cancer stem cells.
This is incredible.
It is exciting. But we do need to remember that this is still early research.
Right.
We need more studies to confirm all of this in humans and to figure out the best doses and any long-term effects.
So cautiously optimistic for now.
Exactly. But I think we can say that ivermectin has real potential to change how we treat cancer.
It really seems that way. This deep dive has taught me so much
It's
about cancer and the amazing research being done.
an incredible field.
Thank you so much for guiding us through all of this. I feel like I really understand ivermectin now and all its potential.
You're welcome. It's been a pleasure. The more we learn about how cells work and how drugs interact with them, the closer we get to new treatments for cancer.
And those new treatments will hopefully be more effective, more targeted, and less toxic. That's definitely a future worth striving for.
It is.
Thanks for joining us, everyone. We'll be back soon with another deep dive. Until then, stay curious.